These are what an immunologist will tell you are the fly's basic immune mechanisms. We've got clotting, which happens immediately following an infection. There's a process called melanization, which produces reactive oxygen and kills microbes. There's phagocytosis, which can phagocytose microbes, and then antimicrobial peptides can be synthesized to kill invading microorganisms. But so I said this is what an immunologist will tell you. And the thing is, when you ask an immunologist, you have to understand they've got their own ecology going on. So here's an immunologist telling you what's going on. They'll tell you what they work on. They don't tell you what their competitors work on. And they tell the story so that they can get funding. That's what they need to survive. And so at my institution, if you were to ask most immunologists what the immune system evolved for, it evolved to reject transplanted organs and to cause autoimmune disease. Okay, that's what it evolved for, and for an immunologist to survive in the funding community, that's what you have to argue. So what we asked instead is, what does a microbe think the immune response of the fly is? And to do this, what we did is we took a library of Francisella tularensis novocyte mutants, where we had transposon insertions covering the whole genome. And we injected this and assayed something like 100-fold coverage of these bacteria. So the way we do the experiment is we take the library, we inject it into a fly, we let it grow for 2 days, and we take it out, and we compare the bacteria we have at the end to those that we have at the beginning, and we ask what has dropped out, because we'd like to know what was required to survive in the fly. And looking at 50 of the mutants we got, we found that about half of them were sensitive to reactive oxygen by testing them on plates with hydrogen peroxide and paraquat. We found that about a third of them were sensitive to antimicrobial peptides, as tested by putting them on plates and hitting them with an antimicrobial whose name I can't remember right now. It's the thing you find in Neospora. And then we find that there are about a third of them that we can't explain with either antimicrobials or reactive oxygen sensitivity. And I find this interesting because we expected to find reactive oxygen sensitivity and antimicrobial sensitivity as things that the microbe needed to survive. But there is a section down here that tells us there's something else, and we need to know what that is. So we wanted to know if we then made mutants in the fly that removed the selective pressure from the microbe, would the bacteria be able to grow better? And that is the case. So looking at reactive oxygen, the way this is produced in the fly is you'll have an immune elicitor that activates a serine protease cascade, activating this enzyme CG3066, and the second one ultimately activating this enzyme phenyloxidase that turns DOPA into melanin and produces reactive oxygen. So if we take our mutants, mutant bacteria, grow them in a wild-type fly, or a fly that can't make reactive oxygen in an immune response, we find that the reactive oxygen sensitive bacteria grow better in the mutant fly. And this is done as a competition assay. So we put mutant bacteria and wild-type bacteria together and then look at their competitive index. So if the competitive index is less than 1, it means the mutant bacteria grow poorly. And you can see that the mutant bacteria grow better when you remove the reactive oxygen stress in the fly. And we can see this with 2 strains. And everything I put on here is significant to at least less than p equals 0.5. We can do the same thing with antimicrobial peptides. We can take the AMP-sensitive bacteria, grow them in a fly that's lacking the signal transduction cascade that turns on AMPs in response to this bacteria. And in that case, with all our mutants, you can see that they grow better when they don't have the antimicrobial peptide pressure. And so what this does is it gives us some tools. We now have some microbes with various sensitivities to reactive oxygen and antimicrobial peptides. And it also makes us really confident that what we think is important for killing microbes is the same thing that the microbes think is important for killing microbes. Okay, so I want to talk a little bit more about this melanization process. We found that non-melanizing flies are sensitive to listeria infections. So if we take a wild-type fly infected with listeria, it dies with a mean time of death around 10 days. If we infect a non-melanizing fly, it dies with a mean time of death about half that. And we wanted to know where the what bacteria this is affecting in the fly, because Listeria can grow both intracellularly and extracellularly. So the way we do this is we inject the bacteria into the fly, and some of the bacteria get taken up by phagocytes, some grow free in the hemocoel. We then inject an antibiotic, gentamicin, into the fly, which kills extracellular bacteria, but not intracellular bacteria. And then we grind the fly up, plate it out, and ask what bacteria are present in it. What we find in a wild-type fly is that if we add gentamicin, or we don't add gentamicin, we end up with the same amount of bacteria in the fly, suggesting that all the bacteria are intracellular. But in the mutant fly, if we add gentamicin, we end up with exactly the same amount of bacteria as we do in a wild-type fly. So the number of intracellular bacteria is constant. But if we don't add gentamicin, we end up with about 5 times more bacteria, suggesting that this immune response is needed to clear the extracellular bacteria in this infection. So that's what melanization is doing here. It's clearing intracellular bacteria, but it doesn't affect, it clears extracellular bacteria, but it doesn't affect the intracellular ones. So we thought, we understand this. And my student went on to just make sure we really understood this and infected the fly with different pathogens. And it was really exciting because every day when she came in, she got a different result with each pathogen. So it was exciting. So if we map this on the map, what we saw was that Listeria goes down here. We see a decrease in health and an increase in pathogen load. And so we say that that suffered from a loss of resistance, and it's probably slid down the curve. But we see 5 other phenotypes with 6 different bacteria. So for example, when we remove this immune response, the fly survives better and kills Strep pneumoniae better than it would have, okay? And then we end up with some strange ones. So E. coli is not so strange. You remove the immune response and nothing happens. Okay, that's easy. But with Burkholderia, you remove this immune response, you end up with more bacteria, and the flies live just as well. So they've got a higher dose of bacteria, which we think means that they actually have to have higher tolerance than they did originally. Likewise, these Ephicalis-infected flies have a lower number of bacteria, but they live the same amount of time. So their tolerance must have dropped. Okay, so you've got balance changes in resistance and tolerance in these flies. And you can't see that unless you measure both colony counts and survival at the same time. Okay, so I want to give you more examples of this. So when fly people look at immunity, one of the main things you'll hear about is we talk about antimicrobial peptide induction. And this is typical of the data will show you that when you infect an animal, you go from untreated, where you don't have expression of antimicrobials, to treated, where a lot of antimicrobials turn on. Okay? But the whole array looks like this. So the antimicrobial peptides come on. There are a lot of other genes that come on. And there are a lot of genes that get turned down. And we don't talk about these very much. And we got excited about them because we found a mutant with the following pattern. This is a mutant in a transcription factor called ETS21C that lacks tolerance to Listeria. So you infect it with Listeria, it drops dead, but the Listeria don't grow any faster. And what I really like about this is that the antimicrobial peptides are fine. Nothing changes. Everything else changes. And so that makes us look at everything else. In particular, I like these genes that normally turn down but didn't turn down. And these are metabolic genes. They affect the mitochondria and the rest of the body. So what we did is we did a metabolomic analysis of the flies. We took healthy flies and sick flies, sent them off to find out what changed in small metabolites. And we get data that looks like this, where we get 250 metabolites back from a mass spec analysis. The overall picture is that not much increases. There are a couple of major decreases we see. Carbohydrate levels drop in the sick flies. Free fatty acid levels drop in the sick flies. And there's a story about mitochondria that we're working on that just isn't quite complete enough to talk about yet. I'm going to focus on a little story here having to do with how flies pee and talk about allantoin and uric acid. Okay? So, when flies get sick, what we find is that levels of uric acid drop about 50%. And the levels of allantoin rise about 5 or 6-fold. So I should tell you, uric acid is the nitrogen waste for flies. Flies, when they break down purines, when they break down protein, they make uric acid, and that's what they excrete. Humans, we also